I would say go ahead, Darren.
Okay, sure. Um, Hi everyone, my name is Darren. But before we start, I just want to say that I actually cannot see the chat. So if you type something, I might simply have to answer that at the end of the talk. So if you have some questions, you just speak out and let me know. Um, and I'm happy to be here to share with you the work we have been doing in the past few years at BioCars, which includes probing transient actually disordered protein structures with time-resolved X-ray solution scattering, as well as some theoretical development in an attempt to interpret BioCars data. We all know how difficult it is. So this talk will be divided into roughly four parts. So first one is going to be introduction of using X-ray scattering to probe transient solution structures, which I believe everyone is pretty familiar with. The second part is an experiment, which is we use temperature jump to trigger a model system. In this case, the alpha lactalbumin. We trigger its unfolding process. And after the signal processing, we have these, characteristic signal that we want to explain. So that leads to the third part, which is theoretical development, where I used MD simulation and we incorporated the X-ray scattering signal into that. And with that, we will be applying to the same system, applying this method to the same system, and then we will do a structural analysis of that process. Okay, but even though I just kind of want to define the scope of this talk, which will be limiting to the protein system. We know that protein can assume different kinds of structures and different states. Well, we know that when it's synthesized at some point, the protein may be unfolded and it folds to its native state to perform functions. Sometimes the protein will be in a kind of strange environment, such as you have a raised temperature, you have a different pH in the cell, you have different ionic strengths, or even the membrane can exert mechanical force on the protein, which makes it a misfolded protein. And we know that these states are the precursors to the aggregated proteins, which are known to be, well, fairly toxic to the cell and sometimes induce or be the trigger of the neurodegenerative diseases. We know that the protein can be in these different states. If we map it onto an energy landscape, we can see that the native state is just one of the minima the protein can be in. In particular, the partially folded states, which are the precursors for the amorphous aggregates, is of interest. And in our group, we're interested in knowing how the native state becomes partially folded state. Specifically, we want to know what part of the protein becomes unfolded or disordered, and how does that happen? So we also know that these high energy or relatively higher in energy states, they may have just a short lifetime, and which means they are transiently populated. And to follow that transientness, we have to sort of survey the timescale of motions in the protein. So we know that there are larger protein reorganization, such as the structure consolidation and sometimes the reorganization of the topological structure. They can be fairly slow. Sometimes you can use, say, a rapid mixing to sort of track these processes, but there are fast processes such as local structure formation, especially the alpha helix that can form really fast. As you can see, the time scale is down to about 100 nanoseconds. And there are also hydrophobic collapses, which are sometimes the first steps of the protein folding process. And in order to be able to track these with the speed, we have to have a really fast flashlight, as can be illustrated by this artwork, where a bullet is shot through it when an apple, and if it not were not for the really fast flashlight, we will not be able to freeze this in time and see clearly what's going on. Okay, so that brings us to why we're here at BioCars, because we benefit from the time resolution provided by the synchrotron and X-Files if you bring this to a different instrument, right? And the reason we're working with X-ray scattering is because it's a method to probe the size and shape of the particles in the solution as I guess people know pretty well here. On the right here, I'm still listing a summary of different properties you can use X-ray scattering to probe, such as, for example, in the middle, we have this reference scattering pattern as a function of scattering angle, and also the intensity, the intensity as a function of scattering angle, right? And if you somehow make the protein larger, then you know all the features will go shift to the low Q part, which reflects larger features in the system. If you make the protein aggregate, you see the low Q part really shoots up in intensity. That's actually how we detect radiation damage, right? On the lower right, you see that if you make the protein unfold, you can perform tracking analysis to get a fingerprint, which shows you that the protein becomes a random network. And if you're interested in the tertiary or even secondary structure changes, you look at the difference in intensity, which encodes basically the entire encodes the structure change kind of in a convoluted way. And on the left, this is the very simplified experimental setup at BioCars, which I believe people know. We use a laser pump from the top, inducing some sort of environmental change. Well, in our group, we're also interested in not only using the optical pump to induce, say, a photo absorption transition process, we're also interested in other kinds of excitations, such as we've been using IR pulse to increase the solvent temperature in a few nanoseconds, which heats up the protein. We've been also using UV pump to convert the photo acid, which changes the local pH value in about 20 nanoseconds, which provides a pretty unique way to change the protein or a peptide structure. Right, and after some time delay, you use the X-ray probe to probe the structure change, and you register that onto the detector. One thing, as you look at this real image on the detector, is that you see that it's a ring. So the signal is isotropic, which means that you lose quite a lot of information, which results in a low resolution reconstruction, as we'll talk about in the later slides. Okay. And I want to first introduce the model system we use. This is bovine alpha-lactalbumin. It's a pretty small molecule or protein, if you wish. Its main function is actually to regulate the production of lactose in this case, cow milk, but it works equally in human body. The function itself is not of interest, but the fact that it can assume a molten globular state. People have been actually studying the molten globular state of this protein for quite a while. They have a lot of descriptions associated with that. Some seem pretty contradictory, like this molten globular state can be compact and not really unfolded. They still exhibit a considerable amount of disorderness. It has less alpha helical content, but it still has an alpha helical signal when you measure this in the CD spectrometry, right? And it's really hard to imagine a structure that matches all of these, being able to probe the entire structure and then gain some confidence by doing that. So this is what we're going to do. In our group, we have developed this really interesting flow cell, and the artwork is credited to Dennis Leshev, who was a postdoc, an extremely talented postdoc at our group. So I just think that because this is about our talk, it would be interesting to talk about this flow cell. And this flow cell is made of an aluminum nitride piece, which is an excellent heat conductor and allows us to control the temperature at the capillary point. We have a Peltier element beneath it, which provides active heating and cooling properties, or actually functionalities. And below that, we have an aluminum water chiller, which doubles as a sort of a mount to the goniometer and rather not that good temperature control in case the Peltier breaks down, right? So this flow cell allowed us to perform many different experiments, from insulin to ubiquitin to the bovine alpha-lactalbumin that I'm going to talk about. But before we actually do the the time result study, we decided to first look at the equilibrium unfolding. We thought that, okay, because this protein has a fully unfolded state and a fully folded state and a state in between, maybe we can simply see it using equilibrium unfolding. It turns out we cannot. So in the middle, these thinner lines are scattering patterns as a function of temperature, and the yellow dot is the fully folded scattering pattern. The red dots are fully unfolded ones. You can use these two scattering patterns to fully model all the curves in between without the need of using a third state. And as you can see, as temperature increases, the fraction of unfolded protein grows in a two-state fashion. So we thought that, okay, so if we cannot see the state but it exists somewhere in the conformational space, we might be able to see it using the temperature jump experiment. So we applied for the mean time, we did the experiment. After a lot of signal processing, we arrived at this pretty complicated signal evolution. So what you're looking at here is the protein-only response at the low, and we're only showing the part where there are detectable difference. Okay, and you can also see there are two shades in this panel A, where the Q1 and Q2 are arbitrarily defined regions. The reason we're choosing these regions is to show that they have distinct kinetics. So if you think about that, if you only have one state or one intermediate state and they grow and decay in a concerted fashion, no matter what range you choose, you will always get the same kinetics. But here we're clearly seeing that there are distinct kinetics, which means that there are intermediates, and indeed there are 3 based on the SVD analysis. So we then used global analysis to kind of require that the signal or the time-independent signal changes in population based on the kinetic model. And our kinetic model looks like this, where the first path is from folded state to an intermediate state to first unfolded state. And the second path is from folded state to the second unfolded state. Okay, and after we did this analysis, we've arrived at the panels C and D, where the C is showing the population of different states, and the D is showing the different scattering pattern associated with that state. If you didn't quite understand that, imagine that if you simply multiply at every time delay C with its respective signal in D, and then you sum that for that time delay, you will basically recover A, which is the signal evolution. Okay, so panel C is just something that we use to fit the data, but panel D encodes the structure change of the protein for these states. We have these signals. What do we do with them? Well, you could say that, okay, since the forward scattering seems to be positive, you see that the protein increased in size or in surface area. You see that there are dips for all the states. So you can say that maybe the alpha helix content is lower, but how do you know where in the protein these helices disappeared or unfolded? That's a separate question, and that's going to be a harder thing to analyze, right? So for a miraculous two full years, Lynn allowed me to pursue this goal of trying to find where in the protein the alpha helices unfolded. But before we do that, I want to sort of do a short survey of structural analysis tools people are using here. For example, if you use ab initio modeling, you can have a low resolution representation based on the scattering pattern, which in this case is the PYP, right? And you can see that there are a tail or a head. I'm not sure the proper terminology here that shows you there are structure changes in the protein. And then you can, and also you can use, I believe Tom's here listening to this talk. This is a great work. You can actually estimate the electron density map based on the scattering pattern using I believe the iterative retrieval algorithm to get an estimate of the electron density. And these are great methods. But for me, actually, I'll talk about that later. And then the second category of the structural analysis tools would be to do a rigid body fitting where you have, for example, a lot of alpha helices, in this case, the hemoglobin, right? You move these alpha helices until the scattering pattern matches the data. And that is also a very good method because you have only this amount of degrees of freedom to work with. So it provides a pretty good, you can kind of nicely fit the data. But in my case, I don't really have these luxuries because, for example, because we're trying to model a disordered protein. So in this case, I have a changing amount of helices that I'm working with. So I can't really use rigid body fitting. And I cannot really use the ab initio modeling because my protein is becoming a soup or a large, like my protein ensemble is pretty diverse. And like every protein molecule will probably be well fit into electron density map, and that will not allow us to discern whether it's a good fit or not. So instead, we have to use MD simulations where we move all items based on the force field. We'll talk about the force field later because it's a pretty, you can think of it as a prior belief where the actual interaction energy of this protein is the prior. And then we use the X-ray scattering signal as the likelihood to refine the structure. So we decided to pursue this route. And here is the 30-second primer of the MD simulation where you have your molecules, your protein, the solvent, and the ions to neutralize the charge. You simulate these molecules in the computer system where all particles will follow the Newtonian dynamics F equals ma, where the force is actually determined by a force field, and there are many different force fields that you can use. Usually they will contain these 5 terms on the right, where the bond, angle, and dihedral, these are the bonded forces, and there are non-bonded forces such as van der Waals interaction and also the electrostatics, right? These are fairly standard parts of the MD simulation, but if you have data and you know how to convert that into external force or potential, you can use that to refine your structures. Before we look at the formulas, we can actually look at this figure on the bottom. So for example, you have this in this dark blue, you have this free energy curve you have this free energy curve, and that favors unfolded state. You started with the unfolded state, and you really want to know what the data drives it to. You then simply add this external potential derived from data, which in this case favors the folded state. Effectively, it just kind of penalizes the unfolded state such that it's easier for you to, for the system to cross the unfolded state, cross this barrier and arrive at the folded state, which also fits the data. Right? Can we do this in the X-ray scattering? For sure, because the Debye formula is a function of coordinates. Even if you're trying to consider the hydration shell, it's still a function of coordinates. So we can exactly calculate the scattering pattern for every cube, and due to that, we can construct this chi-square potential. It simply is the spin constant times a chi-square, basically. And on the right, you see the expression looks like you supply the experimental difference. You then calculated the deviation from, you calculate the deviation of the calculated difference to the experimental difference, and then you weight that by the standard error or standard deviation of the measurement, and then you sum that, and that you then get this this potential. And once you have the potential, you can take the negative gradient, which by definition is the force that you act on each atom. And the derivation of this is detailed in, I believe, Professor Westenhoff's group work, and also in our work. It's pretty essentially the same, but we take care of the hydration shell. This is a long expression, but if you look at the figure on the upper right, you see that it's basically just one vector for each atom. We spent about 6 months trying to make this computation fast because there are 3 loops. One is for Q, the other one is for J, and and then it's for every atom. So there are a lot of terms to compute, but we also noticed that this is great for GPU computation because this is a very ordered matrix-like operation. So we spent quite a while trying to parallelize this computation. We noticed that for every Q, you can actually calculate the force separately. And given a Q, you can also calculate the force for every atom separately. But these are technical details. All right. So after that, we were able to speed up the calculation of scattering patterns as well as the forces by about 7,000 times, which brings down the say for the ubiquitin. If you use one CPU, you have to waste 10 seconds to calculate the scattering pattern. If you use the GPU, you bring it down to about 3 milliseconds. Well, this is not a fair comparison because you can, in principle, use more GPU and say MPI to make it faster. But this is just to show how fast you can calculate the scattering patterns with GPU. So the program flowchart is pretty simple. You use the MD engine of choice, actually in this case the NAMD, to simulate the dynamics for T steps. And at that point you pass the coordinates to the GPU device, which calculates the surface area, which will be mentioned in the next slide. And then using the surface area, you calculate the form factors. And then with the form factors, you calculate the scattering pattern and also the force. You pass that back to the host, which is the MD engine. And you repeat this simulation while applying the external force until your data is fit, or your simulation converges, or you're happy with the result. Okay, and one word about the surface area, which is a fun byproduct of this project, is like this. So we know that there are hydration layers around the protein because of the partially charged surface atoms in the protein, and that attracts weakly the water molecules. So we know that there's a layer around the protein that has a slightly higher water density, and that has been a major topic in terms of I believe in the biophysical community for decades. But in this case, we simply took the implementation from the previous group who developed the Fox web server, which allows you to to calculate the scattering pattern. By basically looking at for each atom how much surface area in terms of fraction is available for solvent access. Sorry, there was a typo in the topic in the title. It's solvent accessible surface area. I'm sorry about that. It turns out that this method can be easily parallelized, so we can kind of move all the computation onto the GPU. It also allows you to visualize the actual hydration shell of a protein, which looks like this. This is for cytochrome C, which is one of the protein I worked with. As you can see, the protein atoms and the hydration shell is clearly there. It's a pretty cool thing. Just want to share with you. But this also allows us to take care of the hydration shell by correcting the form factors of the surface exposed atoms. Okay, we developed this code, we decided to put it to tests. There are two test cases. One is the lysine arginine ornithine binding protein. This protein is great because it has two crystal structures that we can use to create a manual difference scattering pattern. One is its hollow form, the other one is the apo form in red. Okay, we want to drive the hollow form to the apo form. So we simply input that scattering difference, and then we try to whether it can drive the structure to another one. And as you can see, as you follow through the trajectory, you see that the root mean square deviation to the hollow form is getting higher because we're driving it away from that, and that to the apo form is actually lower. I'm going to do a very risky thing, which is to play a video over a Zoom chat. I hope you can see it. If you cannot, it's basically the cyan, which is the simulation itself, is going from the yellow structure to the red structure. I hope it works. Okay, I hope you can see it, but it's basically what's going on here. Okay, and then the next test case is a slightly more complicated one, which is the deca-aniline folding. So this is a model system in the computational chemistry society, where it has two stable structures. One is the random coil, where it has a very long end-to-end distance at about 28 angstroms, and it has a very low alpha helical content because it's just not a helix. On the other hand, there is another stable structure, which is alpha helix, and it has a very defined end-to-end distance at 14 angstrom, and it has a very high alpha helical content. We want to drive the random coil to the alpha helix. And in this case, you can see that the difference signal, the difference force scattering is actually negative because the protein has lost surface area, and that's why we're seeing a negative signal here. But we also put this signal into the simulation, and what I'm going to play is again a video. And on the right, you see the alpha helix actually goes up as it approaches the helical structure. And the X-ray scattering associated potential should be minimized because that's the objective function sort of that we're trying to minimize. So I'm going to play the video. As you can see, this peptide collapses to form this half helix. And then it stays there actually for a while until it finally forms the full helix. It's a pretty satisfying clip to watch. And it also provides us some insight. For example, I can stop there, okay. For example, as you can see, the protein actually collapses. It does not really curl up and form a helix. And that's something we actually have in the agenda to improve, because right now we're trying to model the force as pairs of, as the force between pairs of atoms, and that they are simply dragged toward each other, which is not that physical. If we could map this onto the torsional angle of the backbone, that would be a much better way to model the structure transition. So just throwing out some thoughts here. But with these two test cases, we're able to show that our code works. And we decided to simply apply that to our alpha-lactalbumin system. Okay, so let's review the scattering difference that we got from the signal analysis. They are shown in red here. So for the I state, we have this red trace, and the same goes for the U1 and U2 state. In this case, we're trying to find structures that are balanced, and that gives, sorry, let me go through this again. So we also pretty much did what we did in the past slide, where we input the signal into the simulation and then let it run until it finds some structures that fit both the MD force field and the experimental signal. As you can see, this refinement is not perfect. We argue that it's actually because of it's trying to trade off between the force field and the X-ray scattering potential. And think about that, if you simply don't apply the potential, it would be, you are simply running a conventional MD simulation. But if you simply increase the potential of from the X-ray scattering to infinity, where in this case you're effectively just dumping the force field, you can force the structure to give a perfect fit to the experiment, but then the structures you sample may not be physical. So we're trying to find a trade-off between these, and this is one way to look at the signal, the refinement. On the other hand, you can also look at the free energy surface. In this case, we are projecting all the structures onto the free energy surface constructed by the radius of gyration of the protein, as well as the alpha helix score of the protein. We included the folded protein here, which has a defined radius of gyration at 14 angstrom, and also a fairly high alpha helix score. And we noticed that for the I state, it's actually just a slightly expanded state with a radius of gyration at about 16 angstrom and a lower alpha helical score. And the U states, they are unfolded states, so they have a much larger radii of gyration, right? What's really interesting is that when we are doing the refinement, we did not actually put the kinetic information into that, which means that the refinements are done independently, and the simulation does not know that the signal we input into the I state is the intermediate. It just knows it's a state. And the fact that we were able to find out that the I state is an intermediate through this refinement is pretty remarkable. Okay, and once you have this, the good thing about MD simulation is that you have all the snapshots and you can start to project them onto different structural properties. For example, you can project them on by looking at the solvent accessible surface area change, or you can look at the secondary structure change for the entire ensemble of refined structures. Here I'm showing you, for example, for the I state and delta SASA, this is showing you where the protein actually gained surface area. And in this case, it's pretty clear that the surface area gain is solely from the C terminal, which is the residues from 90 to 110. Okay, and for the U1 state, you see the surface area is gain is pretty much everywhere but in the middle of the protein. Compare that to the U2 state where you have a more substantial exposure of the core, and simply with the surface area change, you can already tell that the unfolding pathways can be kind of categorized as one is the unfold from surface pathway, where you peel off the C terminal and then you peel off the rest of the protein surface residues. On the other hand, the other unfolding pathway is more like an explosion from the middle of the protein, where it expands a lot and then becomes more solvent exposed. If you look at the change in the secondary structure, what baffled us is that the I state actually lost a lot of helices, but it's really strange because it still is very compact with a radius derivation of about 16 angstroms. So we're trying to find out why, and we noticed that we can also show the structures of these show the sample structures of the state. It turns out that the structure looks like this for the I state. And we thought that this might be the molten global state based on the radius derivation and also the look of this, this ensemble structure. We can then use this figure to examine or to scrutinize the different descriptions associated with this state. Is it compact? For sure, it's pretty compact. Actually, you can align most of the backbone onto the crystal structure. Is it not really unfolded? Yeah, for sure, because most of the protein are actually there. But it's disordered, as you can see from the shifts of these backbone traces, right? It's like shifting a lot. It has less alpha helical content because this alpha helix that unfolds completely. But if you want to measure the CD spectrum, we think that it's still possible that you get some CD signal because you still have this helical arrangement for some of the helices. Okay, so this is actually the last part of my talk, and there are a lot of improvement that can be done to make this refinement protocol better, but that will be passed to the next grad student, Adam. I'm not sure Adam is here or not, but yeah, I'll be helping you. And finally, I would like to thank group and especially Lynn, who gave us great freedom to try out these different things. For example, jump, pitch jump, and the theoretical development. And also, I would like to thank people who helped us with the experiment at Northwestern, Dennis Doloff, Arnold, and Professor Kevin Kostedt, who helped me with a lot of the simulations. And thanks to the BioCars crew, who are tremendous in helping us with the experiment, especially Irina, who's our beamline scientist, and local contact, and also the funding source, because without them, we will not be able to do any experiments. And finally, thanks Irina for arranging this seminar series that we can discuss. And I look forward to hearing other people's, hearing the audience's feedback, and also hearing your talk in the future. And finally, thanks for your time and your attention, for your listening. I'd be happy to take any questions you might have. I'm not sure if there are questions in the chat, but just let me know. I'll try to answer that. And yeah, that will be the talk. Thanks.
Okay, my understanding that we can ask Darren directly, or we can put the questions in the chat. So is there anybody who can ask a question? Okay, Thomas?
Hi, that was a great talk, Darren. Really enjoyed
Thanks.
it. So I have a quick question. So you use mostly sacks. I'm wondering if this is also accurate enough for wax, and also related to the wax, do you have to do, which I guess is also for the sacks, do you have to do ensemble calculations, or do you do this with just one trajectory?
Right, so for now we're actually using one trajectory, but ideally you will want to do a replica exchange sort of simulation, and then you optimize or you refine the, so you will have, like, for example, 8 trajectories, and then you calculate the scattering pattern from all 8 trajectories, so you average them, and then you refine that average signal against the experimental signal. If you increase the number to infinity, you're basically recovering the real experimental condition, which is computationally impossible, but that would be a more physical way to do that. In terms of if you want to extend this to wax part, one thing that comes into my mind is that you will have to correct for the polar dispersity of the beam itself, and then you will also, I'm not sure how in the wax part the hydration shell will affect the signal itself. So that's something we're still looking into. But I would imagine that the hydration shell will be of less impact in the wax part. In fact, I believe there are people in the audience who did small molecule scattering. I believe they just calculate the scattering pattern in vacuum and go with it. Yeah.
Great, thank you.
Did I miss something? Yeah.
No, that answers it. Great, thank you.
Thanks.
I also have a question for you, Darren. This is
Yes.
Irina Kashleva. Are there different classes of proteins which will work better for these simulations? You have quite an experience. You run these simulations for several different systems.
Right. So for now, I imagine because that heavily depends on the implementation or the model of the hydration shell. So for example, if you have a different solvent, in this case, I'm talking about insulin, which in our case, we did the experiment in 20% ethanol, which will definitely have a different effect than the pure water or the buffer, right? And in some cases, we use a high concentration of guanidine hydrochloride that will also affect the hydration shell itself, because the guanidine hydrochloride will preferentially bind to the protein surface, so that would change the hydration shell a lot. There are multiple models regarding the hydration shell. What we're using is the most primitive one, where we single-handedly sort of assign a higher water density based on the surface area every atom has. But in reality, all of the atoms will have a different interaction with water. For example, the carboxylate group will have a very high hydration shell content because it has a full negative charge there. But for example, the lysine group will not, because it has a long aliphatic chain, and that will actually rather avoid interacting with the water. So what I'm trying to say is that the hydration shell is atom type dependent, and there are models to address that. If that can be done, then this method can be applied to different molecule systems for sure.
Okay, thank you.
So Darren, might I ask you a question? This is Philip Anfenrud speaking.
Hello.
So a beautiful talk. You've taken this quite far. And I like what you've done with the salvation shell. We think quite a bit about that as well, how we can account for the scattering changes that we see as a function of temperature by looking at that salvation shell. But I have a technical question for you. When you say that you can get, up to 7,000-fold speed up by using GPUs, I've never gone that route yet, but you've made me think maybe I should be thinking a bit more about that. But when I think about that problem, I thought the biggest benefit you get is you have, as I understand, hundreds of cores available in a GPU graphics card, whereas you have a handful of cores on your normal CPU. I would have expected then to have on the order of a few hundred times improvement. How do you get to 7,000? Of course, you can code things poorly and have a big dramatic difference, but from a hardware perspective, I don't see how you get up to 7,000.
I see. So in a GPU, we have, at least for the NVIDIA GPUs, we have, so we actually have blocks and threads. So in reality, a GPU has, in this case, I believe the GPU I work with have, GPUs I work with have 56 separate multi-processing units, where each unit has about, I think, 1,000 blocks, and for each block you can initiate about, in my case, 256 threads. So if you multiply all that, which actually gives you about, like, I think 500K threads that can be done at the same time. But then you also have to calculate the clock speed difference between a GPU and a CPU, which is about 4 to 6 times. So overall, you can actually get a theoretical boost in efficiency of about 100K, which I'm still quite short of. Yeah, so I think you're missing the threads here. Yeah. Sorry, I'm not sure if that's clear. I wasn't prepared for answering this technical question.
So maybe I've been thinking about it incorrectly, because I was thinking about, like, cores. How many cores are available?
Right.
And each core is clocked at a certain rate. So I'm not sure if I understand how you say this clock speed is so much higher on a GPU, because on my
it's actually lower.
2.3 gigahertz. And so in terms of clock speed, and it's a 64-bit processor, so you can, if you code things right, you could in principle process 64 bits at a time, but a lot of times you're processing smaller chunks and you're still only getting the value of that from one clock cycle. So the CPU,
actually the clock speed on GPU is actually slower than the CPU. So for example, for an Intel CPU, will have like 4 gigahertz, right? And for an NVIDIA GPU, will have about 1 gigahertz clock speed. So that's actually slower. But then you have so many more blocks and threads. And if you were able to make, this is called single instruction multi, I forgot the last thing, single instruction multi calculation or something, instruction, then you will be able to boost the efficiency a lot.
Okay, I think it's clear I got to do a little bit more reading up on this to get a sense for what you really mean by blocks and threads, because at the essence, you have a processor that's going to be doing floating point math, and there's a finite number of those, and they all have to actually pull in information before they can do the clock cycle. And so anyway, the fact that you have substantial gains in performance is eye-opening.
Thanks.
That was very nice. So again, thank you for your time.
Thank you.
If I may chime in here, Christopher Hallen from DCU.
Hi, Croy.
First of all, Darren, that was a very beautiful talk, extremely informative, enjoyed it very much. I just wanted to chime in from the small molecule community and saying that also the salvation shells and the salvation cage is of extreme interest to us right now. Mm-hmm. And it's something that we're working very hard to figure out. And some of the methods that you have outlined here will come in very useful in that work.
Nice.
So thanks a lot for that.
Yeah, I think Tom was referring to like really high Q part, like up to, I don't know, 6 inverse syndrome. I'm not exactly sure, but I believe at that point that the cage signal will not be that high, right?
You're absolutely right, and we're going to use that to sort of fix the solute structure in the optimizations. such that we have fewer free variables when trying to optimize the salvation cage.
Right.
Thanks a bunch again.
Thanks.
I have also a question for you, Darren. In addition, is this software publicly available?
Oh yes,
right
it's
now?
on GitHub.
Okay.
So yeah, if you're interested in that, you can take a look at that. Currently, it runs only on NMD because NMD somehow has this arbitrary force module that allows us with any kind of force. Yeah.
So what type of software you have to download to run it from the practical standpoint, except your software from GitHub? None?
Well, you need a GPU device to run it.
Right. Yeah.
And then this is written in C, so it's not. You need a NVIDIA compiler, but that's just a C compiler you can download. Apart from that, you need a working copy of NMD, and I think that's all.
So this is a reasonably straightforward installation for those people who would like to try it out, right?
Right.
Hello, may I ask a question? Martin Nielsen from the Technical University of Denmark. Very nice presentation, congratulations. It's also a very nice paper, I really enjoyed it. But now you have shown this slide 12 for some time, and I'm really curious because you have a gradient in the calculation of your forces, of course, but I always thought that when you put gradients in this way, in fitting, then you also run a risk of having numerical instabilities. So I know this is a rather technical question, but how do you ensure that you don't have problems with numerical instabilities? Do you need to do some kind of averaging, or?
Yes, in our case, this is the raw form of the force, but in this case, we actually do averaging, but what we're averaging is actually this calculated scattering intensity part, because what we noticed is that if you have a little change in the structure, you get wildly varying scattering pattern. And if you simply use that instantaneous snapshot to calculate the force, you'll get a widely fluctuating force. So what we do is actually averaging the scattering pattern. Yeah, and in terms of numerical,
is that averaging in Q, or?
It's averaging in time.
In time, okay.
So basically, when you simulate things, you have a number of snapshots. You cannot simply take one snapshot and then calculate the force based on the scattering signal it gives, and then calculate the force, because that is going to fluctuate a lot. And so what we average over a number of snapshots before that using a characteristic memory. So basically it's exponentially decaying memory into the past, and that gives a more stable estimate of the calculated scattering pattern, and that will also lead to a more stable force that's acting on the protein. Yes. And that's one thing, and the other thing is we try to make the spring constant as small as possible, such that if we were to have any numerical instability, it will be corrected by the MD simulation itself. I know it's not like a perfect solution, but it has been working.
Very interesting. So, but do you then get? I mean, now you have shown time results here, and you are averaging in time. Is that compatible, or do you still retain does it limit your time resolution? I think is the question.
I think not really, because when we're sorry, I clicked right again. The characteristic memory of this averaging, which we use in the computation, is about, I think, 1 picosecond. So it's actually, sorry, that was not right. It's 50 picosecond. So it's still fairly short compared to the actual time resolved experiment. I mean, if you were to have an experiment where you have a time resolution of down to femtoseconds, then you might have to use a replica exchanged version of this code, where instead of averaging over time, you average over the replicas you have.
I see. Okay, interesting. Thank you very much.
Thank you. Irina?
Yes, does anybody else have a question? No? Okay, it looks like there are no more questions. I would like to thank you very much, Darren, for his beautiful talk, very well thought and encompassed a lot of experimental and theoretical work. And I would like to thank you everybody for coming to this first Biocars Zoom seminar. I hope that we will continue, and then I hope to see you all during our next seminar, maybe in October. If you have interest to give a talk at our seminar, please let know Vakita, Rob, or myself. We also can reach you asking for a presentation. Thank you so much, everybody. Great
Thanks,
to see you
everyone.
here. Thank you, Darren.
Thanks, Irina.